Thank you for the introduction, Bob, and thank you everyone for still being here in this late time of the day. I really appreciate it. So we're going to switch gear completely with my talk because, like Bob said, I'm focusing on education, and hopefully by now your interest has been piqued about all of this awesome data, all of this information that's been produced by Jack, Andrea, Joanne, and everybody who presented this afternoon. And all of this information is part of this online course that we are launching that is called Principles and Application of Genetics and Genomics to Improve Animal Health. I want to start my talk by saying that I didn't do that by myself, not by any stretch of the mind. It's a collaborative effort. Two universities really collaborated and received some part of the grant. It's Iowa State University, but also University of Minnesota, and we worked together to design that online course. But there are also all of those other institutions that participated in the creation of that online course, and you can see it's really international. All of those people behind me that you may recognize, some of them have talked today, or some people are going to talk after me. Some people, the research has been mentioned yesterday during our keynote speaker, Dr. Addison, mentioned gene editing. That's Kristen's work, and she contributed to the course. Dr. Nick Serrao is in the committee that worked together to do the conference today. So really a lot of really bright people that contributed and shared their area of expertise for this course. So I want to give you a little bit of background, and we talked about this really major grant called Genetically Improving Resistance of Pigs to PRRS virus infection. And the main goal of the grant was to identify, validate, and transfer genetic markers and biomarkers associated with superior response to and better ability to cope with PRRS infection. And I think you've seen that a lot of data has been produced. And one of our specific objectives with this grant was to develop and implement strategies to educate industry stakeholders on the role of host genetics. So that's the idea behind this course. All of us are trained as veterinarians, may remember our years at school, and maybe genetics was not the most emphasized area of the curriculum. We are really well trained on infectious diseases, but we may lack on the genetic side. And so we thought it would be a great idea to design this course and really emphasize this genetic part. But also, if you are interested in genetic selection and genomics, and you've been out of touch with the field for a few years, well, you may fall behind because it's moving at a fast pace. And that's also the other part we wanted to develop with this course. So our overall goal is to train veterinarian, geneticists, industry stakeholders on basic concepts of genetic selection and genomics and how to apply them to improve animal health. So how did we do that? Well, to do that, we use what we call in education a backward approach. And so what we mean by that is we have goals, and we have a set of goals that we are trying to achieve with this course. So by the end of the class, hopefully, the learner will be able to synthesize epidemiological and biological characteristics of an infectious disease, explain the basis of a genetic improvement program and how such a program works, compare and contrast the various genetic selection tools and different approaches and how they can be used to improve health, but also recognize that there are limitations and that genetic selection may have an influence on epidemiology of the pathogen involved. And finally, how we can take all of that information and apply it to PRRS. So to do that, we considered our audience, and because we are we want mainly to work with practitioners and professionals, we knew that this course needed to be completely online. It is also a course that is self-paced, meaning that you start it whenever you want and there is no strict deadline. So if you want to dedicate 15 minutes only during 1 week and then 2 hours another week and then not do anything for 1 week, that is possible. It is not on strict deadlines. The overall content is 8 hours. But what I wanted to tell you is that in online education, when we talk about the amount of content we put online, we have to assume that it needs roughly 3 to 4 hours to be processed by the learner. Mainly, it's like when you're at a conference and you're listening to people talking to you for an hour, you don't register the content right away. You need to go home and process this information. It's the same idea with the online course. The way we design our content, we have videos, but also what we call sliders, which are interactive content with figures as well as some reading on that course, and what we call knowledge check, but you may know them more likely as quiz, to highlight those major concepts. So each and every single one of those quiz, depending on the answer you select, if the answer is wrong, you have an explanation for every single one of those questions. And then the course gives you the opportunity to answer to that question again, and then move on to the following part of the course. So this is the overall structure, the way we design the course. There are 4 major parts. It starts with the introduction to infectious disease biology, and if as a veterinarian, you know that you think you know everything about infectious disease biology. There are also some parts on disease modeling in that module that maybe you may not be familiar with and that could be of interest to you. Then the module 2 is about genetics and genetic selection, and we start really from the basics about DNA and the genome, but we build up pretty rapidly, talking about genetic variation, quantitative trait genetics, and finally the use of genomics in genetic improvement. So now that we talked about genetic selection, module 3 is actually showing how we can use all of those tools and apply them for disease resistance. Part 1 is about the basic concepts, and part 2 is about the impact that genetic selection can have on disease epidemiology, something that one of the presenters mentioned earlier today. And our final module is how we take all of this information from the 3 previous modules and apply it for genetic selection to PRRS. And you'll see in this last part you have research about the gilt model, but also the growing pig model. And in that other technologies, you will find the latest research on gene editing. So this is what the course looks like. It's hosted in a platform called Canvas, and this is the welcome page. Please disregard those gray little boxes here. These are just the design tools that I have when I am in the course, but as a learner, you wouldn't see that. So you can see that on the right-hand side, you have the different tabs, and you can go from one module to the next. Module 0 being the training on how you interact with this online course. And then this is the introductory page for every single one of those modules. It starts with an introduction video that really summarizes the learning objectives of the module and how the module is structured. And you can see my previous slide, I stopped at this part, but we really broke down, and I'm realizing this is too small and you cannot read this. What I wanted to illustrate here is that we really broke down the content into little chunks and pieces of content so that you can stop at a point in the course and then come back to it. And each pieces of content looks like this. So here we have a video that's been developed by Dr. Deckers. And it may look a little bit small, but you can actually increase the size on your own web browser. And we have the transcript for every single one of those videos. This is an example of those knowledge checks that I was talking about. So you can see it's just a regular quiz with question, not limited in time. You can take your complete them at your own pace. And like I said, you have an individual feedback for every single one of your answers. So by now you've probably realized that there are a lot of little parts and pieces of that course, and you may be wondering, well, if I stop at any point during the course, how do I know where I am at? And this is this page called Course Progress, and you can see, well, because I'm one of the designers, they're all checked for me, but let's say, for example, if you stop here, all of these green check would be gray for you, and it gives you an easy way to actually go back into the course and take it back where you left off. So who should take this course? We believe that swine practitioners, we may have to be involved in decisions regarding genetics, could find it useful. If and you were yesterday during one of the sessions and may wondered what the breeding equation was. This is in the course. Some geneticists that may not be working with improvement in animal health may find it useful, researchers in swine health, and graduate students. So for more information on this course, the opening is in spring 2018, and you have this short URL. This web page gives you all of the information that I presented to you today. There are also some flyers that I put on the table in the back, also in the lobby, that recapitulates all of this information. Here is my email address. If you have any specific question, I would be very happy to answer them. So this project was funded by the USDA, and we are very grateful for this.